Right. Good afternoon, my AP students, and welcome back to class as much as we can have it. Last week I was out during homecoming week, so I'll give you guys a bit of an assignment to try to guide you into our next unit, is it's basically unit 3, and we start talking about cellular energetics and kinetics of enzymes. And so we have a few things that we really need to go over in class. And I tried to post you guys just kind of carry you on assignments and AP videos. You know how we use those in the classroom. And I want to bring those up with you now to show you where we were intended to go, where I wanted you to look, so you can kind of go do that on your own. Then additionally, I want to lecture on the content, get that across to you. So I'm going to open up my screen sharing here. We're going to go ahead and get with it. So this is my first video trying to do a Zoom recording. I think it's pretty straightforward and self-explanatory. So hopefully you guys are all looking at my screen right now. And I just want to kind of take you into it. Now I have my AP Classroom already opened up. But again, I always like to tell you where you're going first. So if you check it out, right, the first part of our chapter, the unit that we need to get into, our unit is here, cellular energetics. Look at that top. At a glance, look at our topics. These are the things that you guys kind of get in, all right? And what I really wanted to get through was topics 3.1 and 3.3, really to a degree before I got back. And then from that, we're going to talk about photosynthesis, cellular respiration, and fitness in general. And all these things connected to big idea 1 and big idea, excuse me. Basically, cellular energetics and system interactions. Okay, our mistake on whatever. All right, our focus here is going to be a reminder of enzyme structure. All right, so we needed to review that enzymes are proteins basically that do chemical function. These chemical functions create a biological output for life. All right, and then they work by doing a process called catalysis. So ultimately, you guys are going to need to look at experimental procedures that are aligned to certain questions. That's a scientific skill and task. We're going to do that laboratory work when you guys get back. All right. So all the stuff right here is science practice. We're going to be on that. Then we're going to use some of the experimental scientific practices to predict causes or effects or changes to biological systems by looking at environmental impacts on proteins. About that a little bit, but enzymes are proteins, so they're going to do the same thing. We can use some sophisticated techniques. All right. After we get done with that, doing our AP lab, we're going to move on and do photosynthesis and respiration in a combined lab. Very important that you guys understand those things when the time comes. All right. So again, just to really focus you guys on the things that you need to know. Topic 3.1, our enduring understanding is that living systems require constant inputs of energy information in the exchange of macromolecules. All right, that's from one of our big ideas. Right? The way we're going to do that is we're going to describe enzyme properties. So the structure of enzymes and active sites where they're going to actually interact with molecule chemistry and illustrate that. And then for enzyme mediated chemical reactions to occur, we have to think that shapes of molecules and shapes of the substrate or the protein itself, they have to be perfectly coinciding. So if you can think of anything that can change that, it's going to affect the function. All right. Then catalysis. This is where we're going to get into. Okay. We're going to explain how enzymes affect the rate of biological reactions. We're also going to hopefully develop the understanding of essential knowledge that enzymes are biological catalysts that facilitate chemical reactions in cells. Here's the kicker part by lowering the activation energy. All right, I'll explain that more in a little bit. Then beyond that, what things are going to change that ability for that activation energy to be lowered? Okay, so this is where your lab's really kind of heavy to teach this and reinforce it. So I'll give you some input. Okay, so we have to look at how changes to the structure of an enzyme can change those functions. All right. Also, we're going to look at how changes to the molecular structure of a component in the enzyme system basically change the function 2. And that's going to lead us to explore the topics of denaturation. So basically, when you eat proteins up too hot, they can eliminate the ability to catalyze reactions. We talked about this back when we were discussing macromolecules in our first unit, right? Also, pH, the electrical environment, it's going to change the polar nonpolar interactions of the structure of protein. Okay, but those are things that we're going to explore as we talk. Though, another thing that we're going to need to reemphasize with you guys what pH actually is: concentration of hydrogen ions. But you're not going to have to kind of do a chemical. You're not going to actually have to use area of that kind of stuff. And relationship between pH and hydrogen. And so that's kind of where we are. All right. Then we'll get into cellular energy, photosynthesis. So that said, your textbook correlations for these, right? Kind of last or circle my last pointer here. Highlighting it a little bit. All right. Here's the deal. Concepts 3.3 and 3.4 are the big ones in the textbook. I might have mistyped it in the announcement that I posted to you guys. But basically, if you guys go back to that chapter, we already did the smart kind of exercise. We did the running assignment, and I'm sure it included it. Now we're really going to get you mastered on these concepts. But if you need to review, definitely. Okay. Now, for the previous assignments, kind of guiding you guys, you go to AP Classroom or click on my internet. Here's what you guys need to do. Right now, when you log in, it's going to bring up a window that reflects what you guys look at. What I want you to do, though, I understand Joseph told me having a little difficult finding what to do. My assignments. Click on my assignments, it should bring up all of your active assignments. I have this. All right. Now, this is the instruction kind of way, but go to all assigned, and basically you should see a list of everything I've given you to include those progress checks. All right. So you can go back and look at your old stuff there. Now, what I wanted you to look at, right? This was all just done for completion. I wanted you to use this to kind of supplement. All right. 3.1 video. 60% of you guys watched it. So then you would do the enzyme structure. Okay. So basically, it's 3 questions. Those would be like the 3 questions we did in class. Remember every topic and have some something questions. All right. Then you watch the 2 video and then do another one. 3 video, do the next one. I want you to just try to do it. I don't want you to be afraid of failing. Remember, one of the best ways we can get this across to you well is take that first honest assessment of what you know. Stumble if you stumble, and if you know, good. All right. Then we go fill the gaps and then maps it, and then assessment you much better. Pretty good following this one, right? So that's where you guys are going to go. Like those 2 watch those 3, 1, 2, 3, 3 daily videos. Look at assignments. Go to all assignments. Find those enzyme catalysis, enzyme structure, environmental impacts on energy. You go through and do those. One of you guys I see went through every bit of it. I'm not going to have your student results. And this was passing, right? So we'll get through it. All right, so that brings us on to the next major focus today, what we're trying to do. All right, so for Unit 3, classroom textbook reading correlation. Check. We're done. Now, I want to take you guys over to your text materials from 3.3 and 3.4. Okay. I want to teach you a little bit. So I'm going to go ahead and go full screen here. On the start of 3.3, a bit about enzymes. Enzymes are incredibly important for life to happen. Without it, it wouldn't occur. I know I've talked with you guys about this in the past, but how it happens is what we're going to do. Living systems, they depend on reactions that are left in natural biological conditions. They call it standard biological. All right, and basically atmosphere pressure, 20% oxygen. The standard temperature is like 25 degrees C, but we know it gets hotter and colder on Earth. But these general range of conditions you see on the surface and the biosphere of Earth, that's what would happen. And if we left most of our chemistry that runs our cells to do their own thing in a cup or a beaker or a flask, it would take hours, days, maybe sometimes hundreds of years for something. Can't happen. No way, that's a compelling evidence against evolution. But the way these things really do work, though, is through the process of catalysis. We have catalysts, all right? And if you look carefully at the screen as I'm going, you'll see my mouse pointer. Wish I could make it right. All right, I'm going to highlight points as I go through the topic. Catalysts, all right? These are substances that speed up reactions without being altered themselves. So if they're unchanged, they're basically like machines that work, all right? Like a wrench turns a nut or a bolt, the wrench doesn't get changed, and then it can turn another one, all right? But instead of turning materials specifically, we're bending molecules into shape so that we can change chemistry. That's a chemical reaction, all right? Here's the deal. No catalyst, though, could make a reaction that can't occur on its own. Catalysts are helpers. They make things happen. Most biological catalysts are proteins. Some are actually a basic RNA molecule, like ribosomes. Those are called ribozymes. We refer to most of those. Okay. Now, in order to understand the principles of this, it's not just the stuff. We need to think about energy because chemistry doesn't happen without energy. Just the way it is. Okay. So I want you guys to think a little bit about this. All right. Energy is stored in chemicals. I mean, that's why we eat food, right? And it gets stored in chemicals, maybe from physical things too, like synthesis. Right? Here's the deal. To do most of the things in biology, we need to get that energy. And how do we talk about that energy? Well, we're going to call that stored energy that's available to do work, this thing called free energy. All right. Top right-hand side of the screen, free energy, and is denoted by capital letter G. Wow. The reason for that is basically there's a guy named Josiah Willard Gibbs who did a whole bunch of scientific studies for us and really helped us with thermodynamics. And so this free energy, it's actually called Gibbs free energy. And it's basically the free energy that can be released from chemical bonds from the high state to the low state. All right. Biologically speaking, get out of chemistry and physical chemistry class, all right, where it's really kind of nuanced. In biology class, it just means the amount of energy in the system that's available. So here's the deal. Without a catalyst, that molecule still has that free energy available. All right. What we need to do, though, is get from that high free energy state to a low energy state. How are we going to get there? And why doesn't it just go there on its own? I mean, if it's at a high state already, couldn't it just go on its own? Well, here's the deal. We need to get over a little hurdle. Otherwise, it would have gone on. All right. Now, I don't know if you guys can see the top right-hand side of the screen, the video talking with my hand, but all right, maybe that'll help illustrate a little bit. In order to get to that next phase, you usually have to bump over a hurdle. We call that the activation energy. And it puts reactants, the thing that you're starting with, as the free energy in the system, into a transition state. Okay. Pictures are way better at teaching this. So that's why I changed the slide. Right. So if we take a look on the top left-hand side of this, I want you to imagine maybe a sugar molecule, reactant. It's stable. Sugar just sitting out in the air doesn't change and lose its energy. But if I set the sugar on fire in the presence of oxygen, all right, I can burn it and make carbon dioxide. All right. You can also do this in our mitochondria. So here's the figure representation of the process happening. We have a molecule in a stable state. Hang out just the way it is. If a chemical reaction is going to occur to release some of that free energy, we have to make it less stable by putting energy into it, and then it releases and goes to a more stable configuration. Right. That difference is called delta G. If you take a look at the graph on the left, there's an important thing I want you to take away. The free energy on this graph is higher numeric value for it. Right? Let's say this is zero energy down here. This is some other number energy higher up. We have our starting energy, it's higher. For anything that naturally will just occur, delta G is always going to be negative. High state to low state. The change is negative. Energy always drops. Activation energy from itself, it can, all right, we can heat sugar up like I talked about, literally setting it on fire on a Bunsen burner or any other kind of reaction. They all require a little bit of heat, all right? Now, in living systems, though, it won't work because those reactions would be destructive. They would basically wreck other materials in the cell. Maybe they'll destabilize cell membranes. Maybe they'll denature proteins and do all kinds of damage. So life's way of getting around that, by God's design, I give it credit. Evolution would say basically that these are chemical accidents that came, or is what it is. But basically, the answer to it is we have to use enzymes. Enzymes lower the activation energy by enabling reactants to come together. They bring materials together and react, or they will take a material and break it. All right, or something else more complicated. For every reaction, there's an enzyme that goes. This gets really complicated when we'll talk more about that later when we talk about DNA expression and how DNA enzymes. All right, but here's an example: molecule sugar. All right, in solution, just in water, sugar and water, like my tea drink cup. All right, if I left it in there at standard conditions, one molecule might hydrate in 15 days. I get one to break down into glucose is whatever. Bad biology. That said, all right. But if I put sucrase enzyme in, the reaction will occur less than. You need that speed of time in your cell. So comparing the two situations, all right. Maybe in a normal circumstance, a little bit of heat, all right, is enough to get over if I have enzyme present. If it's catalyzed. Uncatalyzed, I might need much more activation energy, all right? I might have to go much higher up in order to get it started. And that can be disruptive, make it inefficient, or usually it's just, all right? Now, great. We get the energy process, we get the why, we get the energetic, what's required, all right? But how specifically do they work? Well, enzymes are very specific. Each one typically will only catalyze one reaction. There are exceptions to this, exceptions to everything, right? But the general trend that we see, because the molecule fit is so specific, you don't typically have many molecules that will just fit, all right? So let's describe it and diagram. The thing that's going to go into the reaction, energy, the agent that's going to be reacted is called the substrate. They're going to approach an enzyme or approach each other in an environment, and then they're going to bind to a very specific site. This is where those hydrophilic and hydrophobic interactions come into play, polar, non-polar. Maybe you have a polar positive substrate and a polar negative active site, and they will bind each other. All right. That's how it happens down in the molecular. Forces are at play, the design's there, it works. All right. The specificity of how that works comes from the exact three-dimensional shape and chemical properties of that active site. Here's how it looks. We were talking about sucrose and sucrase, right? Well, your textbook authors and the preparers of these slides try to give you a little bit more simple approach to how this looks. All right. You have sucrose up at the top. All right. We have an hexose sugar and a pentose sugar having a 1,3 linkage, 1,3 glycosidic linkage. Stuff is just all right. This particular bond goes that particular way. These back sides right here of the hexose and the pentose, right? They have active sites on sucrase that overlap part of the backbone or this bridge here of the enzyme. So what happens? You put them both into my cups. All right. In that water solution, the two attract each other magnetically. You see over here on the right side of the screen, electromagnetic attraction. They come together and there's water everywhere. So what happens? The binding of the sucrose to these sites causes the enzyme to change shape. When things bind in biology, imagine a blob of sucrase bending down a little bit, putting strain on these bonds, oxygen bonds, oxygen carbon bonds here. exposing it to water. Then water can get split. The OHs and H's go where they're supposed to on these molecules to make them electrically stable. Now the bonds are broken. When those bonds are broken, that releases the strain, allowing the sucrase enzyme to return to its original shape. When it does, it spits out the glucose and fructose, and we have chemistry. So generally speaking, here's how we talk about that. We have the enzyme substrate complex, all right, held together by hydrogen bonding, electrical attraction, or temporary covalent bonding in some cases. Our active transport that we're going to talk about, membrane transport, really comes into play here. We did talk, excuse me, all right? So temporary covalent bonding, if we find some ATP, we can force that change in the protein. It's all about tertiary and quaternary structures, electrical interactions, and bonds making things move by design, right? So here's what it, generally speaking, we have an enzyme and a substrate that come together to make an enzyme substrate complex. And they become an enzyme and product because the substrate, all right? The big takeaway here, the emphasis point, the enzyme itself is not changed at the end of the reaction. It returns to its original configuration, so it can do the same value. That's biologically smart. We want the tool to be able to be used again, okay? Enzymes basically work on that mechanism, all right? And do the chemistry specifics. I kind of talked a little bit about this without intending it, but all right, the mechanism, the way they work. One, they induce strain. They will grab onto a substrate and bend it into an unstable state. Okay, the force of the protein squeezing on a molecule is greater than the force of resistance for stability of the molecule. It's like bending something, like bending a pencil and breaking it in half. Right. All right. Another one. Enzymes can grip a molecule, hold it in place, and turn a site on the substrate that other bonds. We see this happening a lot with enzyme complexes in chemistry. On DNA, when they do replication, grab a piece and stick it together. And because it puts the two molecules in the right configuration, it lets reactions occur. Whereas otherwise, it would just be randomly bouncing around inside the cup, inside the cell, right? Another, they can add specific functional groups. Basically, we have an enzyme and a substrate, and they'll do that substrate orientation and stick a chemical on it. That's for sophisticated chemistry mechanisms. They're a little above, but that's just how it works, right? The 3D structures of those enzymes are so specific, though. It will usually only bind one or a few related substrates, something we've already heard about, okay? Most enzymes, or many of them, change shape when a substrate binds, and it's going to cause a change. One of the ways that we like to give this as a metaphor analogy is something called catch. All right, here's a good example. All right, I believe, I don't know the name of this particular enzyme. I have to confess it. All right, so check it out. Sorry, I'm back. All right, when we have two substrates coming in, okay, when we have an empty active site, the enzyme itself kind of open and spread, all right, allows more ease of access. Then when we have the binding of one of our two substrates, okay, it causes a configuration, and arrows drawing in, like the material comes into the pocket, catcher's mitt, like a baseball, getting the catcher's in his hand, and the catcher closes down on it, all right? Kind of like that's a switch. When you bind into it, it causes the bending of the shape. It's almost like the muscles in the hand closing the catcher's mitt, but it's like the backbone. Those alpha and beta sheets and stuff basically stretch and move so that chemistry can occur. Pretty elegant. All right. That's it. All right. Some enzymes require other ions or other things binded to them in order to function. And that's going to open the door for us to talk about a very, very important process called allosteric regulation. All right. Let's just get this out of the way first, though. All right. So those things that are going to be binded to cofactors. You guys ever heard the term, maybe, if you've ever heard the term vitamins, These are important cofactors. You ever heard that you need to have zinc, maybe a little copper, stuff like that in your diet? That's because some of your enzymes are metals. Metal ions, coenzymes, those are vitamins, basically. All right, prosthetic groups, basically non-amino acid groups that can bond to proteins. Remember, we talked about protein modification in the Golgi apparatus. That's why we're back here, right? Why we talked about the Golgi apparatus first. Sometimes when we put non-amino acid groups on it, permanently binding to it, it helps it bind to materials. Another big example is the ribosome. When they put together ribosomes in the nucleus, in the nucleolar area, it will bind RNA on it, active binding site, so that RNA can bind to RNA. More on that later. All right. So some of those examples again: iron, copper, right? Coenzymes like biotin, coenzyme A, those carry certain groups. Okay. Additionally, some important ones that you guys are going to learn about, like ATP, NAD, and FAD. These are electron carriers that are going to be photosynthesis and respiration. I'm pretty sure you guys all remember that ATP drives most of our life from biology class. If not, you're going to get a whole lot more. And prosthetic groups. This is just background stuff, guys. Don't memorize. All right. The more you're exposed to it, the better off you're going to be. Right. Heme groups, that includes iron and big functional bulky groups that bind oxygen and electrons and stuff like that. But there are other ones. To get into, it's very, very specific. The big thing, when we have these groups bound to enzymes, they enhance their function. So making them work, yeah, we can talk about that. How well they work, quantify that. And that's where we're really going to get into the meat of what's going on here. Okay. And also what's coming up with our lab next week. I think the rates of catalog. Okay. I would recommend that if you're getting a little bit of brain drain from listening and talking, that you take a moment, clear your head, go sip a drink or something like that. Pause first, obviously. And then come on back, because when I get into this, it's going to be quite math principles. I'll confess, probably not the best. So go ahead and take that break if necessary. And now, all right. So looking at reactions, I want you to think about rates. Think about life and how fast life can work or should work. Now, that's a complicated thing. But I want you to think about simple terms. You guys know the speed range, right? How fast they happen. How many miles per hour, or how many feet per second a bullet flies, maybe. Right? But with our chemistry, we think about how many moles of reactant product per second or per hour per day. And if we're talking about enzymes and their importance of work, that they change reaction rates to make them efficable and workable for life, we need to look at those rates. And if we're looking at rates and looking at speed, this sounds a little bit like physics class. And well, it will. All right. That said, there's some considerations we have to take into account. All right. One, oftentimes enzymes don't exist in a huge amount compared to what they work on. It wouldn't make sense. It'd be like having a billion workers and only one product to work on. All right. Then they have a bunch of workers doing nothing. So here's what we run into in cell situations. Usually less enzyme and substrate present. All right. That can change in order to optimize. The cells will sense that change it with biofeedback. But reaction rates can be increased to a point and then they'll level. All enzyme molecules are bound up and visible. All right. If that happens, we're going to run into a situation called enzyme saturation. So the major idea, we talked about these enzymes and what they do. They convert substrates to products. Okay? How well do they do it? How fast do they do it? Well, it depends on how many of them are there and how much stuff is there. Okay? How much substrate. Our graph that we're looking at here, remember, look at your axes first, always. We have the reaction rate, how fast it's going over concentrated substrate, how much. Right? So this isn't a graph of something per time. It's like if we made an experiment where on the left hand we had no substrate, and then at even intervals increased the amount of substrate to some degree. If you're designing an experiment that way, so maybe halfway through the graph you have like 1 nanomole of cellulase, whatever we're working with, right? And maybe a half, 3/4. You know, you're doing repeat experiments, generating a curve, and then making the best fit. Okay. So what are we looking at here? What we see is that in a reaction without any enzyme there, the reaction would occur on its own, and it would happen at the same rate, slowly increasing. You guys see what I'm doing here, or I'm tracing it? The slope of this curve doesn't mean it only goes a little bit faster if you add more material to be worked on in the system without an enzyme. What happens if you put enzymes in? As we increase, where we put enzyme in, and we increase the amount of substrate, all right? Just put some enzyme in and don't change the amount of enzyme, just substrate. You have, maybe a couple 100,000 molecules of it. Put more and more of the substrate in, you're having a huge spike because the reaction can go really fast as long as the enzymes are still available to do work. There's always someone, figuratively speaking, a molecule there basically reaching for another molecule, all right? Up until we get to the point where we have so much substrate in there that all those molecules are overworked, can't get to them quick enough to handle it. That will make a ceiling, basically a limit for the reaction. That's called our maximum reaction. This happens in almost all biological systems. And when we do studies like this in the laboratory, we basically set up 10, 15, however many different conditions in a reaction. Study the reaction with different amounts of substrate. You guys will do that next week. All right. The maximum reaction rate is used to calculate how efficient the enzyme is. All right. So the molecules of substrate will convert to product over a unit of time. We're going to call it. Right? Turnover can be really quick or it can be really slow. All right. So we can have 1 molecule per second converted. Or in some cases, these enzymes are so quick and so good, they can do 40 billion molecules. All right. That's something like, I don't know if you ever heard of an enzyme called catalase. It turns hydrogen peroxide into oxygen and water very, very quickly. All right. That one I'll just bubble out. I can show you, demonstrate. Another one that's really, really efficient is the enzyme complex for DNA synthase, DNA polymerase. DNA polymerase. Basically, the molecule that makes NA, molecule complex, can bring in DNA monomers. Basically, it's a reversal wood chip for how fast the materials come in. And it gives us that nice, pretty shape. All right, it's pretty wild. This is 1,000 per minute. So I'll show you a video about that. So anyhow, all right, those are the basic principles and backgrounds of enzymes and how they work. Okay, I want to recap all that. Now, how they continue to work goes through a process called regulation. All right, enzyme catalyzed reactions. It'd be great to have them going all the time, but sometimes you can get toxic by making product or whatever the case may be. Or there are enzymes that catalyze step process. Okay, so usually what's going to happen is you're going to have a substrate become a product, and that product is a substrate for the next reaction. The next product is a substrate for the next reaction. And it basically forms like a conveyor belt line, kind of like what we do in factories. Right? So there can be hundreds of different enzymes working. Some of them will have interconnected pathways. Like, look at this hot mess, all right? I like to look, this is basically a map of known major biochemical pathways in eukaryotic cells. Okay? Everything from making nucleotides, making DNA, metabolizing carbohydrates, whether we're breaking them down for energy or building them back up, making lipids, all right, those fat molecules for storage, phospholipids for your cell membranes, all production of ATP, all right? Acid metabolism, making all those different types. And honestly, this thing is woefully incomplete. Like, this is just a general map. Hundreds and thousands of different things that come together. Some turn each other on, some turn them off. It's like this complicated intermixing web making raw material. Okay? So one way that we study that, you guys don't have to, it's systems biology. All right, so biologists get together, we look at systems and do reactions, see what turns things on and off, and then we comparatively look at DNA and RNA expression and match whether those make predictions about the mechanisms. It's pretty wild. All right, when you get down to it, oftentimes we use computer analysis to do it, and we just jump right into graduate school and talk about it. So just to let you know, that's an area it's really important for us for understanding of disease, and treatments of disease, and also optimizing my sports nutrition people. If you had a background in systems biology, you would be very, very well attuned to talk about the types of prescriptions that you give to an athlete, to a person to help them build muscle, now drop body weight, that kind of thing. So now moving on to allosteric regulation and how we look at it. Cells themselves, right, they can regulate their metabolism usually by controlling an enzyme. All right, an enzyme is a protein. So protein, how do we control how much protein is there? Well, we turn on or turn off DNA. Okay, so we can turn on the synthesis or turn it off by maybe some of the stuff that it's making, shut down the production of enzyme or inhibit it. Okay, that goes back to DNA synthesis, RNA, and the protein. That's our next move, right? But another way that works much faster by adjusting the activity of the enzyme. That's really what I want to focus on. Okay, so chemical inhibitors. Those are one way that we can slow enzymes down. Okay, there are natural ones and there are artificial ones. Okay. Basically, chemical inhibitors, what they're going to do is they're going to bind enzymes and slow down the reaction rate, usually by affecting the active site, making it accessible or inaccessible. All right. So here are the different types. Okay. Types of inhibition. And this is something I want you guys to pick up on. We're going to see some graphs to illustrate what it really means. The first one is irreversible inhibition. This is where you're going to have an inhibitor covalently bind to a side chain in the active site, and you're going to permanently shut down the enzyme and ruin its function. If it's a critical enzyme for life function, it's basically a deadly poison. All right, insecticides work that way. All right, one of them is DIPF. Okay, and then we have this very, very important enzyme for regulating nerves called acetylcholine esterase. Normal function, all right, is that acetylcholine is this thing that gets released from a nerve cell to another nerve cell to turn the next one off. And in order to turn the switch off, we break down the acetylcholine, all right? And the acetylcholine would go into that active site. But what ends up happening with this DIPF, right, this fluorine atom right here is going to bind covalently and irreversibly, okay, into the site. And it's going to pop that in the active site open, stretch it, and plug it up. From there, now every time that nerve cell turns on, doesn't get the breakdowns, flood the area there in the cell, and it causes hyperactivity of those nerves. If you had this happen in you, it would be the equivalent of like mustard gas, or I think sarin actually, nerve toxin does this kind of thing. I have to check myself on that. I might be speaking out a little bit. But basically, potent chemical warfare agents, that's what people use on each other in history, specifically in World War I. That's what it is. All right. Additionally, carbon monoxide. It's a little bit different. You don't bind so much an enzyme, but you bind up that heme group in your blood cells. You guys breathe carbon monoxide or cyanide gas, you get a permanent binding to that, and it blocks oxygen, and your cells are there. Reversible inhibition, right? That's this one, we can slow down, competitively inhibit. All right, we have something that competes for the active site in the enzyme. All right, here we go. Let's take a look at the right? With competitive inhibition, you have a substrate and a competitive inhibitor in the same place. And the inhibitor can get in there and out, all right, and block the active site, okay? Then the substrate can't bind to it, it slows down, all right? Lots of drugs work this way. Non-competitive inhibition binds at a site distinct from that active site, and it causes the change in shape of function and prevents binding. Same kind of outcome. We have an active site of the same enzyme that would work great for this substrate here and here. The how is what's different, okay? With competitive inhibition, it goes straight from the outside. Non-competitive inhibition, we have another chemical that can somehow bind to the enzyme and cause the enzyme to change. We're going to call that allosteric regulation. You need to know what that means. Allosteric. Steric meaning shape, allo meaning other. All right? So it binds a site other than the active site. The enzyme will change shape, which alters the chemical attraction to the active site, blocking itself. All right? And this is very commonly used in biological and biochemical. All right? All right. Allosteric sites can be modified just like the regular active sites. We can do non-covalent bonding, so it's reversible. You can reverse or covalently bonded too. There are natural pathways where this occurs, and then there are ones where we throw poisons and chemicals out there and it can affect them. All right. So just as kind of a recap of what these look like. Again, we have an inactive enzyme. We can have something activating, right? It binds to that allosteric site over here in the top left. Top left side of the bottom right hand side of the middle. All right, here we go. Bang. It binds into it, the active site. So I'm in products. All right. Another way, we have an inactive enzyme. It has an active site embedded in its core. All right. And we have an enzyme like kinase that sticks a phosphate onto it. Putting a phosphate onto the place where it wasn't before makes it really polar, so it can change shape. All right. That opens up an active site, and we get products. All right, it can be used to activate or deactivate enzymes. And honestly, with that, that really goes into where I wanted to focus with inhibition, feedback inhibition. We'll get to that another time. But a last part, and it went to, I believe it was topic 3.3 in your topic guides and outlines, are things that affect enzyme activity. All right, pH is one of them, and it's incredibly important. All right. Remember, if we have lots of acidic side chain amino acids, they can generate H plus ions, anion. All right. Basic side chains, those can attract H pluses and become cations. That can change the pH environment around them. All right. So basically, an example of it, if we have glutamic acid going down our backbone, all right, if you can imagine glutamic acid attached to amino acids next door. If we have a higher H plus ion concentration, the reaction is basically going to be that you're going to have a proton on this glutamic acid. Tons of protons there. It's going to saturate this environment with this negative. That's negative, the carboxylate anion, the COO minus, the H pluses are going to bind to it because there's a ton of them there. It's just going to attract them, right? And then it's going to become a less hydrophilic form, and that's going to enzyme shape. Okay. Point. Here's the big point. That was just an illustrated example. If it's confusing to you, I can illustrate it to you, draw it up, tell you the whys. What's the big idea? What's the most important? Here's the deal. Remember, tertiary structure, the three-dimensional shape of the protein, is very sensitive to H plus ion concentration. I just changed the pH, I put this acid or some base around a protein in. You're going to chemically do the same thing that you do. Alter protein function equals alter protein biology. All enzymes have optimal pH activity. Okay, it's not always neutral. Okay, temperature can also affect it. All right, if you increase the temperature of enzymes to a point, it's going to increase the chemical activity. We have more energy in the system. If it becomes too high, non-covalent bonds will break. Hydrogen bonds will overcome, and it will activate the enzyme. And those two things that I talked about before are really well illustrated in this figure. Okay, so if the stuff before floor was kind of like bouncing off you a little bit, maybe this will help. Let's look at our two different things that we were discussing. On our top, we have pH. Okay? And on the bottom, we have temperature. We're going to change the temperature. Excuse me. Sorry about that. On the bottom, we're going to change temperature. On the top, we're going to change pH. In all cases here, we have reaction rate being measured, how fast reaction occurs. And then we have different enzymes on the top, general one on the bottom. On the top, we have extremely acidic pH of 1. Concentration of H+ ions would be 10 to the 1. All right, so like 10 molar of acid, which is incredibly powerful. All right, and then all the way up here to pH 12. You might have heard the range going from 0 to 14. All right, so down here we have the enzyme pepsin. Then higher up the pH scale, we have chymotrypsin. Then we have arginase. Let me tell you a little about what these are. These are the enzymes that are in your small intestine. They break down protein. Okay? Pepsin breaks peptide bonds. So if you guys can think about it, the logical order of things, imagine food. You guys ate it and chewed it, right? You swallowed it. The first place it goes to after your esophagus from your throat, all right, is to your stomach. And your stomach is loaded with acid down here in the low pH. Okay? So here's what happens. pH goes from incredibly acidic to very highly acidic. This is unnatural. But the hydrogen chloride that your stomach produces, along with this pepsin enzyme, gets it to the pH about 2. That's the peak activity. So you're going to have your first process digestion of the chicken and steak and any protein you had have peptide bonds that fit into pepsin, right? Then from there, your stomach is going to slowly leak out little bits of that processed digested gas that you guys ate. It's going to take some time. That drops into your small intestine, also from your small intestine, and the pancreas right next to it. You're going to let out a bunch of bicarbonate ions. That's going to neutralize the stomach acid, making it neutral to basic. And what happens there? You also have the release of other enzymes into that ductwork of your stomach, chymotrypsin and arginine. Okay? So here's what happens. As we get into the small intestine, we start neutralizing chymotrypsin work and the increasingly basic environment that's there. You get from neutral to just above neutral. It's at its peak activity. So you get most of your breakdown due to chymotrypsin right at the beginning of your small intestine. Then as we go down, get further in, more and more secretions of carbon ion increase. You ultimately could have a high pH, close to 10. It works best, this enzyme works best at a pH of 10. Honestly, it's going to hang out closer to here. Time, all right, as you move through the small intestine. And from there, you're going to have more breakdown and you have the separation all right, from other proteins. You're going to start stripping the protein down to its monomers so that you guys can absorb it into your intestine. That's the big point. That's a really long-winded illustration. These different enzymes work in one pot. They work through the digestive system. Work at the right time in the right conditions that change pH. Acid environment would affect the backbone of all these enzymes differently in different places. So they're aligned in a sensible way. Okay. Temperature, we just do this generally. Each one of these enzymes actually operates the same optimal temperature, 37 degrees centigrade, 98.6. Okay. Let's pretend that we're just looking at one of them. Okay. If we change the temperature, when we go from very slow to very cold, the reaction rate is slow because the energy inside the molecule. As we get toward the maximum rate temperature, optimum temperature, all right, everything's moving well. Then if we keep heating, we start putting too much energy into the system. You'll notice the rate falls off. Okay. Ultimately to the point that if you destroy the enzyme, you get no activity. All right. So with that, I believe that brings us to the spreadsheet. Yeah. There's a lot going on there. I covered a lot here. I think I've been talking for a better part of the month. Right? What's good about this video setup that you guys can pause me and make me stop. You guys get really bored, not a problem. Or if I talk too quick or something didn't make sense, you can just rewind me. So it's a mixed blessing, all right? So what have we covered so far? The intro, enzymes and enzyme kinetics. Now, there's another caution, right? I'm going to go into this lab and what we're going to be doing next week. So go ahead and take a break. Pause me for a minute, all right? And then when you come back, I'll rehash, and then I'm going to go into our next little bit of slideshow to tell you about the illustrated example of Cellobiase, how we're going to address the problem of energy molecules. Basically make biofuels by studying this. That, pause me for a minute, and we'll get. Okay. Now, hopefully we're back and we're ready and moving on. The next phase of our talk is going to be here, introduction to Cellobiase enzyme. Okay. That real quick. All right. Around here. And what we have now, what I have up on the screen is the slideshow I'm going to post to our talks. All right. And we're going to do a lab where we're going to explore the process basically of doing enzyme studies using light indicators. We're going to be able to apply all that stuff that we just discussed with enzyme substrate complexes and being able to measure it, right? And we're going to do that by looking at specific enzymes called Cellobiase, and it's used to break down the sugar from plant material that normally is completely inaccessible. All right, let me go ahead and bring this. I'll just go slide by slide down the side. I should have opened the PowerPoint. So check it out. This particular slideshow is actually a professional development slideshow given for the teachers, all right, that are going to teach and use this. I'm just going to go ahead and teach from it. This is developed by people that are professionals for both education and in molecular biology. It's part of the reason why I use their lab kits for our classes. All right. Nonetheless, let's get to it. So we've already talked about what enzymes are. Give you some background. We have molecules, basically proteins. They speed up the rate of reaction by dropping activation energy. Lots of different types of enzymes. One thing I didn't really harp on too much in the previous part of the video is enzyme classes and types. I don't want you to try to memorize it. It's just too much, right? But I want you to see, right? Almost all of these enzymes, look down the center of our screen, all right? We have ASE at the end of it, okay? So almost all these particular enzymes, pretty much every enzyme has an ASE. So oxidoreductase. Basically, it's going to oxidize something and going to do oxidation of the adding and taking away. All right. Then we have transferases, which basically transfer specific functional groups to something else. Okay. Hydrolases, hydrolytic reactions or hydrolysis. Hopefully, you guys remember hydrolysis and condensation and hydrolysis. Sorry, no longer. All right, but hydrolysis reactions, hydrolase and lyases, which are double bond reactions, isomerases, which bend the new molecule atoms around on a molecule, ligases, which stick things together. All of them, whatever they do, if you learn the root at the beginning, that basically tells you what it's going to do. So hydrolase enzymes work basically by breaking down bonds between dimers or polymers. So that said, how do they work? Remember, we have the substrate and enzyme, all right? They're going to form together and make a complex, all right? And then they're going to produce a product. An enzyme reaction would normally occur on its own, just might need a whole lot more activation energy if it's uncatalyzed. We have an enzyme with it. Figuratively, remember from our previous study here, you're going to have basically substrate for solution. Substrate will bind to a specific cleft or area through hydrogen bonds or other attractions. And the activation energy barrier is going to be overcome because that substrate is going to bend the molecule, snap it, or do other movements that overcome the energy need. And then we're going to do the chemistry. We're going to have a product. So why are we talking about that? All right. Well, in this particular case, I really want to. And I'm sorry, bear with me a little bit. There we go. All right, so now that we know our background on our enzymes, what are we going to apply? And we're going to use the professional case studying the production of biofuels. Right? Basically, fuels that are produced from a biological source are biofuels. We use them all the time. In fact, we've been using one of the most persistent and important ones in industry, oil, for 100 years now. Oil is technically a biofuel. It has a very long production cycle. Millions of years is what they say. Now, again, that conflicts with our biblical worldview of the on earth. And again, if you put energy into a system, change the rates of chemical reactions. But that millions of years time length is based on geological assumptions. You know that most of our biological assumptions from a natural perspective or an evolution perspective, basically they're 4.5 billion years old. And it took hundreds of millions of years for dinosaurs and biomass and stuff buried under sedimentary rock to be squeezed into oil. Okay. So no matter how you look at it, oil doesn't make itself quickly in nature. That's the important point that any one of us can agree to. So in order to get to it quicker, we need to find another way. And so technology and studies have come about basically where we can move short cycle biofuel, right? Biodiesel, ethanol from starch sugars, cellulosic ethanol. Okay. Butanol is like, it's basically bigger. Ethanol, excuse me, ethyl alcohol, ethanol, which is stuff some people drink it, but you can use it for things. I don't recommend you drink ethanol, otherwise, right? And then ethyl propyl just means it has more carbon on it. Okay, but these are short cycle ones that can be made different ways. All right? And we're going to focus a little bit on getting ethanol from starches and sugar, cellulose, actually cellulose. So here's the deal. You guys remember from biology, we have what we call fermentation, adaptive pathway to make energy. And these bacteria can do that. They can ferment sugar and make alcohol. They don't make as much ATP, but the alcohol still has energy and we can use it. All right. But sugar for food and sugar that's available for like eating is scarce in production. Right. But plants around the world make plant cell walls, right? And those cell walls are made of this thing called cellulose. And cellulose is a lot like starch in that it's a carbohydrate, but it just has the wrong bonding to be useful for enzymes that we can use, right? So we needed to find another way. And one way that we look at it is basically that there are other things in nature that can break down plants. Otherwise, plants would die and never go anywhere, right? Fungus produces enzymes to get to it, right? We want to break this stuff down called cellulose, all right? Cross-linked tubes of it basically between cell wall. They might have other materials mixed in with it, depending on the type of tissue that we're talking about. So if we can get the cellulose out of the bulk material from picture A, we go to picture B, then we have like maybe pulp, all right, broken down wood and plant and leaf matter that's loaded with cellulose plus other materials that can affect our reactions. We want to get the cellulose out permanently if we can. All right, so we have to extract that out. These things, by the way, they're like lignins and tannins and other materials that plants make for color and for structure support. But if we can get those tubes of cellulose unravel them and break them down, then we have long single chains of sugar. And if we throw some enzymes in there, chop apart pieces of it, all right, we can do an enzyme cocktail, basically. Break those down into dimers of sugar. And then we can use fungus and ferment it. And we can make ethanol that can be used, right? So how does that work? All right. Basically, we're going to have like the cellulose itself. We need to break it down. We can either do it with heat, acid, ammonia, or other treatments. All right. Usually, they boil the heck out of the stuff and try to break the bonds down as much as possible. All right. They can use endocellulases to break them into single chains. Then exocellulases, like an enzyme mix. All right. We'll put those in there and we'll chop them down. And then a cellobiase and to break them. Then if I have lots of monomers of glucose, that's really useful. I mean, if we could do this, people, I mean, it's not healthy to just eat glucose, but we could eliminate a lot of our food. I'll tell you that much. So here's how we want to focus this lab, the lab that we're going to do on Cellobiase, okay? We're going to look at a breakdown of it and how it works, all right? So cellulose, if we're breaking it down, we have two sugar molecules basically that are bonded together at this position. We've got, if you go around a molecule, I don't know if you guys remember us from back in our original talk on molecular structure and the chemistry of life, but you go to the carbon that has the most substitution on the most stuff bonded to it specifically, right? And then you can go around it 1, 2, 3, 4, 5, and 6. All right, and it has this linkage here called a 1,4 linkage. Number 1 carbon hydroxide, all right, would be here. And the number 4 carbon hydroxide would be here. You guys don't need to know organic naming functions specifically. I'm just explaining why it works this way. Here's the deal. Normally we have a 1,3 linkage. This molecule will be bonded to this one. Animals can use it. Not how it is. This is what's made according to the DNA in those plants, all right? Through the enzyme process that they use. This is what's out in nature. This is what's biologically available. So how do we get at it? Well, we go to something that can break down new bonds, the 1,4 glycosidic linkage, and we turn to the thing that breaks down nature, fungus, and we get this enzyme called Cellobiase. Cellobiase and a 1,4 linked dimer of sugar, right? Reproduction breakdown, but not refined sugar, all right? And water, we bring it together and get 2 glucose molecules. That'd be really useful. We run into a problem here, all right? I can make this mix of stuff and I can hope that I have plenty of Cellobiase. But how do I know how well this reaction works or if it's even complete? All right? Well, I don't. So what we do then is we turn to synthetic chemistry, chemical techniques. We go to the lab. Humans, we come up with experiments and such that help us do studies of them, all right? And what do humans have to do in order to do this? They have to use their senses in order to make some environments. So how do we get to a situation where you can study the Cellobiase when you can't see it? Okay? Cellobiase and glucose themselves, dissolved in water, they're completely colorless. Looks just like sugar and water. Okay? However, people figured out that if we do a quick chemical reaction, all right, and put this material on, okay, which by the way, this is called para-nitrophenol. Bonded on there like the alcohol of the sugar. We have this alcohol, nitrate, and benzene. What ends up happening is it emits a bit of color, okay? When I put this stuff into water, I don't really see much anything. However, when Cellobiase works on it, splits that 1,4 bond, what would be 1,4 bond, it gives off the para-nitrophenol that was on it. It's still colorless, I can't tell. However, when I want to stop the reaction, something useful can happen, all right? If I suddenly flood my reaction with base, sodium hydroxide, something like that, get the pH above 8.6, 2 things happen. One, the enzyme Cellobiase, down immediately. It won't work anymore. My reaction is frozen. The second thing happens is the bases, all right, the OH ions or hydroxide anions, they cause a little chemistry to occur. They basically make this paranitrophenol yellow in solution because they force some double bonds. Let me show you this. Now, this is way above the scope of any high school chemistry class that I've ever heard of, right? I'm kind of taking you into college organic chemistry to tell you how a lot of these work, but it's just to show you how and why, okay? If you have a pattern of repeating double bonds, single bonds, double bonds, single bonds, double bonds, single bonds, double bonds, single bonds. Whenever you see those kinds of structures existing in nature, what ends up happening is you usually absorb energy and give off color. They'll emit light and color. Most of our dyes, most of our chemical dyes and stuff, that's what this procedure is. So this physical problem. So in recap, if I haven't used it yet, I'd be very surprised. We have a product that we can get some use out of in nature. Take it and do some food processing in the industry. We made a model of the system that we can study with this paranitrophenylglucopyranose. Don't even need to say that. Basically, what's going to happen is you can measure color. Okay? So, taking our model system, all right, treat it with Cellobiase, and then we treat it with base after we're done with our reaction. However we want to study the reaction, yellow. So how can that enzymatic reaction be quantified? It's like this, right? You guys might remember me talking about increasing the amount of run a reaction to measure the reaction rate. Well, a basic solution will basically develop any of that paranitrophenol. It'll start the reaction. So you can look at it visually and see the more product is made, the more yellow reaction is pictured here. Okay? Right? But quantitatively, we can use instruments called spectrophotometers in order to kind of take a look at this. We have one in the classroom that we're going to connect to the classroom computer. Basically, it's going to be like this. You guys are going to add enzyme and some material, make a reaction environment, something called a buffer. Then you're going to run your enzyme reaction. And at specific times to measure rate, maybe you're going to squirt that into stop solution, little bits of it. And you see an increase in color. It follows this thing called the blue. So I'll talk about it. It absorbs more light at 410 nanometers. So there are different ways to do it. The BioRad folks give us our reaction kit like they're equivalent. Kind of different way. Okay. Basically, what we're going to do is we're going to measure the absorbance of light, correlate that to the concentration. Remember, right? But we can connect concentration to color. By doing that, measuring reactions over time, we're going to be able to really examine the dynamics, pH, temperature, substrate, all those things are going to affect it. So everybody in the class is going to measure the reaction rate of the Cellobiase enzyme at a particular temperature. We're going to generate calibration curves based on how much reaction material is there and how much paranitrophenol is present. And that way you can make sure that the machine is working out. All right. Through that, we're going to get a hands-on approach and guide you guys to how to use the equipment and make you a lot more comfortable with the system. Okay. This again is just an introduction to the lab. Right. So everybody's going to do activity one, or even on their own. Right. Then from there, the 5 of them are going to pick by changing temperature, changing pH, changing the concentration of enzyme, concentration of substrate. One of you guys is going to do the same thing as somebody else. We're going to run another experiment we're going to design how you're going to change the experiment. All right, and then we're going to bring together all the lab data. We're going to see what the best temperature, the best pH, the best enzyme concentration, the best substrate concentration, given a little freedom for the best. Then taking your foundational exercise, your second day of lab, all right, where you guys are going to do temperature, substrate. When we come back together, we're going to bioprospect for Cellobiase. You guys are going to run a reaction on tissue sample. What do I mean by that? You guys are going to like basically bring in mushrooms, different types, and you're going to squish and mesh them around. And we're going to try to get some Cellobiase. So here are the activities. The first one, you guys are going to measure the absorbance at 410 nanometers. I'll show you how to do that in class. Right? This isn't something I can just teach you. All right. But we're going to make some solutions. Known amounts of paranitrophenol. We're going to make them all the same volume. We're going to measure how much light is absorbed at 410 nanometers. All right. And what you end up getting from that glitching PowerPoint. But basically what we have is a standard curve. We're going to have like zero reaction having occurred. Final concentration. Then if you guys are doing the reaction rates, what's going to happen is you start the reaction and at different times you put stop solution into your little flasks. Okay. Let's take a look over here. Right. Have your enzyme reaction occurring, and then at time 1, time 2, time 3, time 4, time 5, we leave 1, 2, 4, 6, and 8 minutes. You guys will squirt a little bit of the reaction mix into the stop solution. Compare those with your standard curve. Calculate how much is made. You guys are going to be able to then determine the initial reaction, right? What the reaction was at the temperature in the lab on the day that we start. Then from there, you can measure the amount of the effective temperature. Okay, so when you change the temperature, your initial reaction is repeating that previous experiment at different temperature. Other one, right? We're gonna somebody is going to change the pH environment for the reaction and compare that to the original. Then we can have increase the amount of high concentration, low concentration of enzyme, right? Do a high amount, we would predict maybe the amount of the paranitrophenol will happen quicker. We'll have a higher ceiling because more enzyme. And then the last, how fast is the reaction increase by increasing the substrate concentration? So we'll have the original reaction rate, and you do a higher and a lower one if that's your choice. Example, it's 1.5 micromolar and 0.25 micromolar, different. And then the last, bioprospect. This is going to be like kind of like a quiz grade for you guys to go with the lab, like an extension lab and follow on after that, right? Where you guys are going to in some mushrooms. If you have them growing on a backyard, you got it. You could also find cellulase enzymes or cellobiase enzymes in termites. I don't know if you guys have termite wood in the woods near you. Hopefully not in your own home, all right? And I'm pretty sure none of you guys are going to have yeah, but in cow guts, they have cellulase enzymes because they have bacteria in there. The bacteria have cellulase enzymes. You have to extract the bacteria and get the enzyme out of it. I don't have the stuff for it. The enzyme, or excuse me, fungus is the best way. All right. Some mushrooms are the ones that biological rich for food. You guys can go buy them at the grocery store. All right. Some attack plants, some do shit explosion. composition, right? Some are associated with plant roots, all right? I would avoid, if you guys wanted to do porcini or chanterelle mushrooms, those are neutrophilic, basically help the plants. They don't really digest cellulose that well. What shiitake, morels, mushrooms, and honey mushrooms definitely quite parasitic for plants and they will degrade cell walls. So what we're going to ultimately do on that last lab, I'll go over this again with you. This is a procedure I'll go over after we're done. So now I'm going to go ahead and back. So with that, I wanted to just go ahead and reach out and say thank y'all for hanging in there. We went over a lot of material today. This is a very long lecture. And I'm hoping that you guys gleaned quite a bit of information. Half of a unit in one sit down. This would be like an extra review for you guys. As you close things out, but never the most important thing is the concept. That said, I'm going to push this up to the Google Drive and YouTube.